Hello, my name is Dr. Pramod Kumar Agrawal, and I'm one of the general physicians. And I also have research background, so that's why I thought it will be interesting to talk about COVID-19 from a perspective of a biologist to a clinician, and from clinician to a biologist, in the sense that from bench to bedside and bedside to bench. To talk about COVID-19, I'm going to talk in this talk about 15, 16 minutes, basically divided into five simple questions, simple W questions. Well, we'll start is what is COVID? Who is eligible for testing? Where is the sample taken from? How is the sample processed in the lab? And when to rely on the test results? So let's talk about number one. What is COVID? Just try to remember there are three terms which are interchangeably used. The most popular is COVID 19. There is a term called novel N coronavirus. And third is a big one is SARS CoV 2. I will go into them in a chronological order. But historically, just to understand where the COVID started, or this disease we're seeing now as a pandemic started. It started in late December in Wuhan city in China, which is in Hubei province, where they saw their patients presenting with atypical pneumonias, and then eventually they found it was caused by a different virus which we never ever seen before, and which was at that time was called as novel coronavirus because it was a new coronavirus causing those presentations of pneumonias. Then in February 2020, WHO coined the term called COVID-19. COVID stands for coronavirus disease. 19 is actually from 2019. And just to aid in clinician to talk among themselves and for the ease of understanding and generating and distributing informations. Eventually, the third term came, which is called SARS-CoV-2. That's after a lot of virological study, it was found that that virus or the COVID-19 was caused by a group of virus, beta coronaviruses. However, it's a new coronavirus, different from previous SARS. That's why it's called CoV-2, so coronavirus 2. At the moment, unfortunately, about 2.2 million people are affected with COVID-19 and more than 150,000 deaths. What are the symptoms of COVID-19? From Chinese and Italian data, it has been shown that the most common symptom is fever. Other could be fatigue. Also, people complain of loss of appetite, general body aches, and diarrhea, nausea, and people can also present with loss of sense of taste and smell. There are different spectrum of COVID-19 itself, and thankfully, about 85% of patients have only mild disease. 80 to 85% will have a mild disease. And then about
10 to 15% will have a severe disease, and about 5% will have a critical disease, which will involve an organ failure, for example, respiratory failure leading to kidney failure or heart failure. Of the people who will have a severe disease, about 2.4% will die. So the case fatality rate is about 2 to 3%. So on the one hand, there's a big spectrum, but because it is going to affect so many people, we have to limit the number of people who are severely affected at one time. That's why it's all about social distancing, hand hygiening to flatten the curve. Now that we know what is COVID, the second thing is who is being tested for. So just to understand, and from the COVID spectrum, that there are few risk factors which can happen, which can affect the person more with severe disease, and they occur in patient with people with previous comorbidities. For example, people who have pre-existing high blood pressure, diabetes, cardiovascular disease, kidney problems, lung problems. So those are the people who are more vulnerable to be affected and eventually die from COVID. So that's why the cocooning came into place to safeguard those people, to protect those people. Now, testing, the number two is who is being tested. So testing is done. The WHO recommend that we should test as many as possible. However, we also know that there's a shortage of the kit. There is an intense labor force required. That's why different national guidelines and protocols are in place. The place where I work in Ireland, we have decided that we will test anybody who present with either new fever, cough, or shortness of breath. Not only that, we'll also test for the contact person who has been tested, who has been in contact or close contact with somebody who has been tested positive. And thirdly, healthcare professionals who has been exposed to COVID patients without proper PPEs, which is personal protective equipments. So those are the people who are being tested. Now that brings me to the third part of where is the sample taken from if somebody is deemed eligible. And it's very important to understand what coronavirus, where does it reside? It actually resides in the respiratory tract. And it binds to a receptor called ACE2 receptor, which is called angiotensin converting enzyme 2 receptor. So if you think logically, it will make sense to test somebody for the coronavirus presence from the lower respiratory tract samples. But that is not always easy and not possible because it requires intubation or it requires putting a tube into the throat and taking the sample, which is a risk for the procedure itself and also the risk to the healthcare profession. So you would see that most of the tests are done from what we call oropharynx or nasopharynx. WHO recommends that we should test the sample from the oropharynx and the nasopharynx and go into the viral transport medium in the same tube. And that will increase the yield. And it's very important to have a proper sample collection. This is interesting for the clinician because they might not be the first person, they might not be in the front line to collect it. So WHO recommends that it is collected in a synthetic swab because wooden swab or cotton swab may degrade the virus itself. So the synthetic swab goes into the back of the throat, stays there for a couple of seconds so that it absorbs, and then it goes to the nose and stays there in the nose for a couple of seconds so that the virus is absorbed on the surface and it goes into the viral medium. In the medium, it can stay in the normal fridge temperature, so 2 to 4 degree for 72 hours. And if there is a delay, it should be put into the liquid nitrogen. It should be left below minus 70 degree. So when the sample is collected, it is transferred to the lab. And that brings me to the fourth part. How is the sample being processed or tested in the lab? So the coronaviruses are basically single-stranded RNA viruses. So that's why they're processed in a process called reverse transcriptase PCR. What happens there is they are taken into aliquots and then with an enzyme called reverse transcriptase, a complementary DNA is made. And that DNA is processed through cycles after cycles, typically 30 cycles, with a process called polymerase chain reaction. And then that sample is read in the fluorescent uptake. And if there is a detectable label with the control, the person is positive for COVID or the SARS-CoV-2 is detectable. So this is what happened for now. So that the sample is collected, the patient is suspected of COVID, the patient population is selected, the patient is tested, and then it goes to the lab and the lab is processed. Once it's processed in the lab, it is reported.
It is either negative or positive. It's not reported as negative or positive. Sorry, it is reported as virus detected or not detected. So since there are so many steps involved, there is possible of error. And now that brings me to the fifth, when to rely on the results. So from the Chinese data, we know about one third of the samples can have false negative results, which means although the patient has the virus, but it's not either sufficient enough, or there has been error in collection, or it has been degraded, or there has been error in the lab itself that it has not been detected. What does that mean? That means that about 1 in 3 persons can still have the COVID or the disease or the virus, but it's not detected. That's why it's recommended that although if there's a strong clinical suspicion, either different tests should be done or patients should be self-isolating so that you can break the chain of the transmission. Now, when I mentioned briefly about different tests, there is another test where we test the immunological test, where the IgM antibody is being tested in the person's blood. And the Chinese have shown that IgM combined with RT-PCR has the highest yield, about 99%. So there is about 1% of false negative in that case, rather than 30%. The IgM is very interesting in the sense that is IgM is now being used as a therapeutic, what is called convalescence plasma therapy. What happens is somebody who has recovered from COVID, they have made their antibodies, protective antibodies against coronavirus, and that is being taken out, processed, and given to somebody who is critically unwell with COVID-19. And it has shown some promise. However, further studies needs to be done for its feasibility and efficacy. Now, let's analyze this concept of false negative in one third of the patient. It's kind of very interesting and important to understand that logic, that one third of the patient can still have the disease, have the virus, but can be falsely negative. So let's think of a patient who had symptoms of COVID and she was tested positive. She self-isolates herself and thankfully recovers because she only had a mild disease and she recovers fully. And because of the local policies, before she goes to work, she's asked to have a test done. And the test is done and she's thankfully cleared. The test is negative, so the virus load is not detected. She goes to work, she does all the social distancing, but somehow she redevelops respiratory symptoms with fever, headaches, shortness of breath, and cough. And then the clinicians suspect that she could have COVID-19 and decides to test her. And this time she's tested positive. So far she has 3 tests done. One was positive, second was negative, and thirdly positive. And then you might be thinking the 3 scenarios could have happened in her case. The first scenario, that she was never negative, it was just falsely negative, and we are reassured. And third time we are detecting the same virus as the first time. Second scenario is that she cleared her virus, but then she got reinfected. Or third scenario is that she cleared her virus, but the immunological response was not protective long enough to help her to protect against second reinfection. So that's why it's important to understand the concept of false negative. And then the protective antibodies are still debatable how long they are going to remain in the body protective. And this remains to be seen if people can reinfect it with SARS-CoV-2 again or not. So that brings me to the end of the lesson. And I think it will be helpful for future generations who will encounter COVID-19, and it will hopefully this lecture will help to understand COVID-19 and its biology and clinical manifestations. So if you have any questions or comments, please do not hesitate to leave a comment on the box below. And also, I have added some references, which are references from the Chinese study, references from CDC, WHO. I hope you'll find this talk useful, and I really want to take this opportunity to thank all the frontline workers for their dedication and hard working. At the end, I would like to re-stress this: stay home, stay safe. Thank you.